Hello everybody, welcome back to my Knowledge Academy channel. I am Mithu Dey, Public Health Nursing Officer. It is the high time to resist against some fatal diseases just like the coronavirus. So we need to improve our immune system. Our immune system is a very complex network of the cells, organs and the tissues. Together, they help our body to fight the infection. WBC plays a vital role in our immune system. If our blood vessels is damaged, then the primary functions of the platelets is to prevent and stop the bleeding. WBC and the platelets together, they help the body to fight infections and the injury. We should know how the blood cells, that means the WBC and the platelets are produced. So today's topic is the production of WBC and the platelets. WBC, that means the white blood cells. White blood cells are one of the vital blood component, also known as the leukocytes. Shape of the WBC, round and the irregular in shape. These cells are the colorless as they don't have any pigment but can appear as a very light purple to the pink color when examined under a microscope and colored with dye. They make up around 1% of the human blood and size varies from 12 to 17 micrometer in diameter, and the average lifespan of the leukocytes can be anywhere from a few hours to the several days, and the average lifespan of the WBC are 12 to 20 days, after which they are destroyed in the lymphatic system. Platelets, also called the thrombocytes, and they are just like the small plates in the non-active form. Our blood vessels will send out a signal when it becomes damaged. When platelets receive this signal, they will respond by traveling to that area and transforming into their active formation. To make contact in the broken blood vessels, platelets grows the long tentacles and then resembles like a spider or an octopus, that means the irregular shape. These small plates, that means it is an non-active form, and these are all in the active formation. These are all the octopus-like appearance, so it is the active formation. Size of the platelets 2 to 3 micrometer in diameter, and it lacks the nucleus, so it is called the anucleated cells. And it is the small cell types in the blood and can only be seen under the microscope, and colorless cell fragment in our blood. And the primary functions of the platelet is to aid in the blood clotting process. And platelets has 5 to 9 days of the lifespan. It is a picture of the hematopoiesis, and we all know that hematopoiesis is a process by which all types of the blood cells, that means the RBC, WBC and the platelets, are produced from these hematopoietic stem cells in the bone marrow. And this is the hematopoietic stem cells, and hematopoiesis is a tightly regulated process of the blood cell formation, and short-lived mature blood cells are replenished on a daily basis by the hematopoietic stem cells, and hematopoietic stem cells are the stem cells that give rise to all the blood cells, that means the RBC, WBC and the platelets, and this process is called the hematopoiesis. Before going to the leukopoiesis and the thrombopoiesis, we should have some knowledge regarding the stem cells and hierarchy of the stem cells. So let's see what are the stem cells. Stem cells are the body's raw material, and stem cells are the founder cells for every organ, tissue and cell types of the body. Stem cells from which all the other cells with the specialized functions are generated. Under the right conditions in the body or the laboratory, stem cells divide to form the more cells called the daughter cells. And when the stem cells divide into two, one daughter cells goes on to make the more specialized cells and even give rise to the several different cell types, and the other daughter cells remain as the stem cells are ready to produce the more stem cells when they are needed. Only stem cells has this versatility. So the basic characteristics of the stem cells are the self-renew or the self-renewal, and the differentiate into a specialized cell type, that means the differentiation. The distinct property of the stem cells is the ability of the unspecified cell, that means the stem cells, to transform into a specialized cell in the body. This is known as the differentiation. By the symmetric division, stem cells remains in the stem cells, and by the asymmetric division, progenitor cells originated from the stem cells. Some examples of the human cell differentiation. This is the stem cells, and all these are the specialized cells: muscle cells, fat cells, bone cells, blood cells, nerve cell, epithelial cell, immune cell, and sex cells, and so on. Hierarchy of the stem cells. Blood stem cells or the hematopoietic stem cells, they are the multipotent stem cells. They are able to give rise to multiple cells within a lineage. These are all the multipotent stem cells, and these multipotent stem cells are derived from the pluripotent stem cells, and pluripotent stem cells are the capable of developing into many different cell types, and these pluripotent stem cells derived from the totipotent stem cell, and totipotent stem cells are capable of dividing and developing to form a complete and a mature organism. Hematopoiesis is also known as the hemopoiesis and describes the process of the blood cell formation. All the blood cells are derived from the initial pluripotent stem cells, that means the PPSC, which gives rise to the colony forming units or cells. These colony forming units or cells further differentiated to give rise to their final stage of the development, where they became the various forms of the blood cells. The production of the RBC is called the erythropoiesis, and the production of the WBC is called the leukopoiesis, and production of the platelet is called thrombopoiesis. Erythropoiesis was discussed in the circulatory system part 1. Who have not seen this video till now, please go through this video. Link is given in the description box. It is a multipotent hematopoietic stem cells. After cell division, these cells remain as the stem cells, and these cells goes down one of two paths depending on the chemical signals received. These two lineages are separable at the progenitors level. Progenitors. What is the meaning of the progenitor? Progenitor means the direct ancestor, an originator or a founder of the future development. মানে পূর্বপুরুষ, ম্যাচিওর ব্লাড সেলের পূর্বপুরুষ হলো এই মাইলয়েড স্টেম সেলস or the myeloid progenitor cells. Blood cells are categorized as the myeloid stem cells and the lymphoid stem cells. These myeloid stem cells undergo differentiations to produce the precursors of the platelets, erythrocytes, and the WBC and the monocyte. WBC, that means the granular WBC and the monocyte. These precursors are the megakaryoblast for the platelets, proerythroblast for the erythrocytes, and myeloblast for the granular WBC, and monoblast for the monocyte. On the other hand, lymphoblast produce from the lymphoid stem cells, and lymphoblast is the precursors of the T, B lymphocytes and the natural killer cells. Here myeloblast, monoblast and the lymphoblast. But what are the blast? Monoblast or the lymphoblast. But what are the blast? In biology and the medicine, the suffix blast refers to the immature cells and the very earliest cell known as the precursor cells. Blast give rise to all kinds of the specialized cells, that means different specialized cells. And myeloblast, here myeloblast give rise to white blood cells, and the monoblast, monocyte, and lymphoblast give rise to the lymphocyte. Here monoblast, myeloblast, lymphoblast are the precursor cells, and myeloid stem cells and the lymphoid stem cells are the progenitor cells. What is the main difference between the progenitors and the precursor cells? Progenitor cells are the multipotent and differentiate into several types of the cells. On the other hand, precursor cells are the unipotent and differentiate into a particular type of the cells. WBC not only originate from the bone marrow, they also originate from the lymph nodes and the spleen. There are the primary lymphoid organs and the secondary lymphoid organs. Primary lymphoid organs include the red bone marrow and the thymus. They create the special immune system cells called the lymphocytes. And secondary lymphoid organs, it includes the several types of the lymph nodes and cervical lymph nodes in the neck and axillary lymph nodes in the armpit and inguinal lymph nodes in the groin. And spleen and Pierce patches also are the secondary lymphoid organs. Pierce patches. What are the Pierce patches? Pierce patches are the grouping of the lymphoid follicles in the mucous membrane that lines of our small intestine. And lymphoid follicles are the small organs in our lymphatic system that are similar to the lymph nodes. Regulation of leukopoiesis. Certain factors regulates the process of the leukopoiesis. These are cytokines. Cytokines are the small proteins that are crucial in controlling the growth and activity of the blood cells and other immune system cells that helps the body's immune system and inflammatory responses. And this is the cytokine producing cell. After getting the stimulus, this cytokine producing cell releases the cytokines or produces the cytokines. When released, these cytokines produce the signal to the target cells and to do its job. This is the cytokines. Which cytokines are in hematopoietic family? Number 1, interleukins. Interleukin. Interleukin is a protein in humans, sometimes also called the colony stimulating factor. That means the CSF. And there are various types of the interleukin such as interleukin 1, 2, 3, 4, 5, 6, 7, etc. And it has a specific role in leukopoiesis. And the interleukin 3 regulates the growth and differentiations of the hemopoietic progenitor cells. Next, the CFU-GEMM, colony forming unit or cell. A colony is simply a group cells that form the hematopoietic colonies, so called the colony forming unit or cells. Represent a stage of the hematopoietic differentiations between the hematopoietic stem cells and more terminally differentiated cells that are erythrocytes, granulocytes, monocytes and the platelets. CFU-GEMM. It stands for colony forming unit, granulocytes, erythrocytes, monocytes and megakaryocytes. It is a colony forming unit that generates the myeloid cells. CFU-GEMM cells are oligopotential progenitor cells for the myeloid cells or the myeloid lineage and also called the common progenitor cells or the common myeloid progenitor cells. And these progenitor cells produce the precursor cells, and these precursor cells also produce the mature blood cells. That means the granulocytes, basophil, eosinophil and the neutrophil, and also the erythrocytes, monocytes and the megakaryocytes or the platelets. Next, the colony stimulating factors, that means the CSFs. Colony stimulating factors include the GM-CSF and GCSF. Colony stimulating factors or CSFs are the glycoproteins that binds to the receptor proteins on the surface of hematopoietic stem cells and thereby activating the intracellular signaling pathways that can cause the cells to proliferate and differentiate into a specific kinds of the blood cell, usually the white blood cells. And these colony stimulating factors such as the GM-CSF, it stands for granulocyte macrophage colony stimulating factors, and it is a glycoprotein, thus function as a cytokines, and it is a white cell growth factors. It stimulates the stem cells to produce the granulocytes and the monocytes. And next, the GCSF. GCSF, it stands for granulocyte colony stimulating factor. It is same as GM-CSF, but it stimulates the stem cells to produce the granulocytes. Next, the erythropoietin, which is needed for the RBC production, and thrombopoietin, that is the TPO, which is needed for the production of the platelets or the thrombocytes. So let's see finally the regulation of leukopoiesis. Hematopoietic stem cells produce the common myeloid progenitor cells under the influence of the interleukin 3, GM-CSF and M-CSF. And these hematopoietic stem cells also produce the common lymphoid progenitor cells under the influence of the interleukin 17. And these common myeloid progenitor cells produce the megakaryocyte erythroid progenitor cells under the influence of the interleukin 3 and stem cells factor and thrombopoietin. And granulocyte macrophage progenitor cells also produce from the common myeloid progenitor cells under the influence of the GM-CSF. These megakaryocyte erythroid progenitor cells produce the erythrocytes under the influence of the erythropoietin, and it produces the platelets under the influence of the interleukin 11 and thrombopoietin. And these granulocyte macrophage progenitor cells produce the myeloblast under the influence of the interleukin 11 and thrombopoietin, and it also produces the monocytes under the influence of the GM-CSF and M-CSF. And these myeloblasts produce the granulocytes. Granulocytes, that means the neutrophil, eosinophil and the basophil or the mast cells under the influence of the GCSF and GM-CSF and various types of the interleukin and stem cells factors. And monocyte produce the macrophages under the influence of interleukin 6 and stem cells factor and GCSF. And these common lymphoid progenitor cells produce the T lymphocytes, B lymphocytes and the natural killer cells under the influence of the various types of the interleukins. This is a picture of the hematopoiesis, but I want to tell only leukopoiesis and the thrombopoiesis because I have already discussed the erythropoiesis in my previous video. So let's see at first the stages of the leukopoiesis. We all know that this is the pluripotent stem cells, and pluripotent stem cells has the two lineage. Number one is the myeloid stem cell lineage and number two is the lymphoid stem cell lineage. These myeloid stem cells produce the monoblast and those the myeloblast. And monoblast and the neutrophilic myeloblast produce from these myeloid stem cells under the influence of the GM-CFU, that means the granulocyte macrophage CFU. On the other hand, eosinophilic myeloblast and the basophilic myeloblast produce from these myeloid stem cells but under the influence of the eosinophil CFU and the basophil CFU. And here the megakaryoblast produce from these myeloid stem cells under the influence of the megakaryocyte CFU. On the other hand, these lymphoid stem cells produce the T cells and the B cells. Let's see the various stages of the granulopoiesis, that means the production of the neutrophil, eosinophil and the basophil. There are 6 stages of the granulopoiesis. Number 1, the myeloblast. Number 2, promyelocyte. Number 3, myelocyte. And number 4, metamyelocyte. And number 5, band form, that means the juvenile stage. And number 6, the segmented neutrophils, that means the mature neutrophils. In the first stage, myeloblast. Here, high nuclear to the cytoplasmic ratio and immature chromatin with the prominent 3 to 4 nucleoli are present. This stage is common to all the granulocytes. Number 2, the promyelocyte. Here, high nuclear to the cytoplasmic ratio but more cytoplasm than the myeloblastic stage. Here, the round nucleus with the immature chromatin and prominent nucleoli are present, and cytoplasm shows the primary azurophilic granules. This stage is also common to all the granulocytes. Myelocyte. Number 3, the myelocytes. Eccentric placed oval nucleus with no indentation. Mature, that means a condensed chromatin and nucleoli are absent. More cytoplasm and less primary granules are seen and more secondary azurophilic granules are present here. And mind that these cells can undergo mitosis up to this stage. And number 4, the metamyelocytes. Kidney bean-shaped nucleus with indented nucleus, but indentation is less than half of the diameter of the nucleus. Here the mature chromatins are present, and here the only secondary granules are present, not the primary granules. Next, the band form, that means the juvenile form. Kidney bean-shaped with indented nucleus, more than half of the diameter of the nucleus, and more cytoplasm and secondary granules are present here. And number 6 stage, that means the last stage, the segmented neutrophil. Mature nucleus divided into 3 to 5 lobes connected by the thin chromatin filaments, and in the cytoplasm secondary granules are present here. Neutrophils are called the polymorphonuclear, PMN, polymorphonuclear, because this neutrophil has 3 to 5 lobes, more than 2 lobes, so it is called the polymorphonuclear. Let's see how the granulocytes produce from the stem cells. That means the committed stem cells. Committed stem cells are the cells that have become committed to a particular pathway of the differentiations. Adult stem cells are committed to becoming a cells from their tissue of origin and can't form other cell types. As for example, adult blood forming stem cells. These are all the granulocytes colony stimulating factors. This means the GCSF. This GCSF stimulates the stem cells to produce the various types of the granulocytes, that means the neutrophil, eosinophil and the basophils, by following those various stages. In the cytoplasm of the granulocytes, at first the primary granules are formed, and then by following the various stages, then the secondary granules form. These are all the secondary granules, and these are the chromatins of the nucleus, and these are all the secondary granules. And this is the process of the granulocyte forming. Monocyte develops from the same precursor as the neutrophil, that is the GMCFU, and these then differentiated into CFUM, that means colony forming unit monocyte. There are 3 stages of the development of monocyte, that are monoblast, promonocyte and the monocyte. At first, the monoblast. Monoblast, it is the first developmental stage after cell has differentiated into the CFUM. Monoblast are the agranular cells, and they resembles myeloblast except for the tendency of their nucleus to be slightly clefted or the lobulated. A typical monoblast is about 12 to 20 micrometer in diameter and has a nuclear to cytoplasmic ratio is 7:1 to 3:1. Cell shape of the monoblast is a round or oval shape, and nuclear shape round or more regular, and nucleoli 1, 2 or even 3 or 4 nucleoli are present in the monoblast, and they are distinct. And in the cytoplasm is a gray and the cloudy blue color, and cytoplasm has the few red granules, and sometimes the vacuoles have been seen. And next, the promonocyte. The nucleus and the cytoplasmic ratio is the 7:1 to 3:1, and cell shape of the promonocyte is a round to oval shape, and nuclear shape indented or the lobulated, more irregular than the monoblast, and nucleoli, 1 or 2 nucleoli are present in the promonocyte, and they are the less distinct than the monoblast. And in the cytoplasm of the promonocyte, it is the gray to the cloudy blue color, and they have the few red granules. Next, the monocyte. In the monocyte, the nucleus and the cytoplasmic ratio is 4:1 to 2:1. Cell shape of the monocyte, that is the round with the smooth edge, may have pseudopod-like extension. And nuclear shape, indented, often reniform or folded, resembling the three-pointed hat, but may be rounded, oval or lobulated. And nucleoli of the monocytes are generally absent but occasionally small and inconspicuous. Cytoplasm are abundant, gray or gray blue color, and may contain fine azurophilic granules, and these granules contains the enzymes and that can damage or digest the pathogens and also release the inflammatory signals. Once the monocyte has entered the tissue, it differentiates into a macrophage. Macrophage originate from the blood monocytes that leave the circulation to differentiate into different tissue. The main difference between the monocyte and the macrophage is that monocyte is the precursors of some of the macrophages, whereas macrophages are the professional phagocytes which engulf the pathogens invading the body. And monocytes and the macrophages are the two types of the cells found in the immune system, and they are considered as the front line of the host defense. Monocytes are the bean-shaped small cells, whereas the macrophages are the irregular shaped large cells. Both monocytes and the macrophages are capable of the secreting the cytokines and the chemokines, which are important factors in the immune response. And dendritic cells also derive from the monocyte, and they are the distinct dendritic cell subset involved in the inflammation and the infection, and they originate from the monocytes, and their activation and the functions may vary upon the stimulation of the circulation. This is the macrophages and this is the dendritic cells. They are also derived from the monocytes. Next, the lymphopoiesis. The development of the lymphocytes is called the lymphopoiesis. Lymphocytes develops from the CFUL, and stem cells in the red bone marrow give rise to the lymphocyte precursors, and some lymphocyte precursors are processed in the thymus gland to become the T cells, and some lymphocyte precursors are processed within the bone marrow to become B cells. Both T cells and the B cells are transported through the blood to the lymphatic organs such as the lymph nodes and the spleen. So, let's see the lymphopoiesis. Red bone marrow. This is a red bone marrow, and here the stem cells in the red bone marrow give rise to the lymphocyte precursors, and these are all the lymphocyte precursors. And some lymphocyte precursors are processed in the thymus gland to become the T cells, and these lymphocyte precursors goes to the thymus gland through the blood circulation, and these T cells goes to the lymph nodes and the spleen through the blood transport. And some lymphocyte precursors are processed within the bone marrow to become the B cells, and these B cells goes to the lymph nodes through the blood transport, and both T cells and the B cells are transported through the blood to the lymphatic organs such as the lymph nodes and the spleen. Thrombopoiesis. As leukocytes and the erythropoiesis, thrombocytes are derived from the multipotential myeloid stem cells, that is the CFU-GEMM in the bone marrow. CFU-GEMM cells differentiate into the megakaryocytes CFU and then the megakaryoblast, and from megakaryoblast, megakaryocytes, and ultimately the platelets and the thrombocytes produce. So let's see the thrombopoiesis. The process of the development of platelets is called the thrombopoiesis. This is the myeloid stem cells, and that means the CFU-GEMM, and these CFU-GEMM cells differentiate into the CFU megakaryocytes, and these CFU megakaryocytes then develops the megakaryoblast, that is the precursor cells of the megakaryocytes, under the influence of the cytokines CFU and CSF and interleukin 3. Unlike other blood cells which undergoes the mitosis in the first few developmental stage, but here once the CFU-GEMM has differentiated into the CFU megakaryocytes, there is no further mitosis, just reduplication of the cell. The CFU megakaryocytes then develop into the megakaryoblast, and this megakaryoblast cell is slightly basophilic in nature and around 30 micrometer in diameter and has a more or less round nucleus, and that is the non-lobed. And from the megakaryoblast then the megakaryocytes develop, and megakaryocyte cell is around 50 to 70 micrometer in diameter and regulated, that is by the thrombopoietin, respond to the thrombopoietin, and this thrombopoietin regulates the production of the thrombocytes by stimulating the CFU megakaryocytes to mature into the megakaryocytes, and ultimately the thrombocyte formation. These megakaryocyte cells is a giant cell, and pieces of its cytoplasm and the cell membrane bursts off to form the thrombocytes. Once megakaryocyte can produce up to 6,000 thrombocytes, that is the platelets. Thrombopoiesis is mainly regulated by the production of the thrombopoietin, and it is mainly produced by the liver and the renal tubules, and it is regulated by a negative feedback mechanism, and it binds to the TPO receptors on both of the megakaryocytes and the platelets. If these are in the high concentration in the blood plasma, then the level of the TPO is kept low and reducing the maturation of the CFU megakaryocytes and the thrombocyte production. So these are all about the leukopoiesis and the thrombopoiesis. Thank you to all.